Hi, and welcome to Reviewing with Mrs. Wages. Today's topic is functions of DNA and replication. So, let's get to it. DNA is an amazing molecule in its capacity to store information. Because DNA contains genes, the instructions for making proteins, it stores the information responsible for our unique traits. Most people know that our genes are responsible for our outward traits, but DNA also stores information needed to control or run cell activities that we don't normally see. Vital cellular activities, such as cell division, are controlled by the information stored in DNA. Another amazing function of DNA is that it can be transmitted from parents to their children during reproduction. When an organism undergoes meiosis, it produces gametes or sex cells that contain a single set of DNA-carrying chromosomes. These cells are used for reproduction only, and when combined, produce offspring that are a blend of parental DNA. Another important function of DNA is that it has the capacity to replicate itself. This means it can copy or duplicate all that stored information. This function is vital to the success of making new cells. If DNA didn't replicate itself, the brand new daughter cells produced by mitosis wouldn't even have enough DNA to survive. So, when does this process even happen? DNA replication occurs prior to cell division during interphase, and specifically, it happens during S phase of interphase. Since DNA is stored in the nucleus, it makes sense that replication occurs there. So why do cells bother to replicate their DNA? When the mature cell is ready to divide, replicated chromosomes split so that each daughter cell gets a complete set of its own DNA. For example, human somatic cells contain 46 chromosomes. When one divides, each of its two daughter cells will also get a complete set of 46 chromosomes, and both are then ready to carry out life processes. As a recap, DNA replication is the copying of DNA. It happens in the nucleus during S phase of interphase, and cells copy their DNA so daughter cells are supplied with their own DNA when cells divide. Alright, so how do cells copy their DNA? It begins with an enzyme known as helicase. This enzyme helps to break down the weak hydrogen bonds holding DNA's base pairs together across the molecule. Once a section of DNA has been unzipped or opened up, a second enzyme then comes into play. Unlike helicase, which breaks hydrogen bonds, DNA polymerase forms new ones. This enzyme connects free-floating nucleotides to each of DNA's two unzipped sides. A single polymerase molecule adds new nucleotides down one side of DNA, while another will add new nucleotides up the other side. Each old side serves as a template for each new side. In this way, DNA replication is said to be semi-conservative. Finally, polymerase proofreads its work in an attempt to repair any mistakes that could lead to mutations. It's a reliable system, but doesn't always work perfectly, and mutations do occur. And it's important to note that this process of replication occurs simultaneously in multiple spots along the DNA molecule. Section by section, the DNA is copied in this fashion until the entire chromosome has been replicated. Let me recap replication. First, helicase breaks hydrogen bonds. Second, DNA polymerase connects new nucleotides to each old side of DNA. And finally, polymerase proofreads each strand. That's all for now. I hope this helped, and as always, happy studying!